Hi, welcome again to Botany 101, General Botany. My name is Angela Nishimoto and I'm your teacher for this class. Okay? Remember last time we talked about fruits, right? We went into fruits. What is it for? It helps a flowering plant disperse its seeds, right? Because of course, a lot of fruits are juicy, they're beautiful, they're succulent looking, makes you want to eat them, right? So us animals, we're pretty programmed for one thing, like insects and birds and others, to help pollinate a plant in getting our food. And other animals, of course, like to eat fruits in order to disperse the plant seeds. So as a video once said, something about that we are actually the plant servants. You know, they are actually our masters in a way, you know? Isn't that kind of interesting? Anyway, so we talked about fleshy fruits, right? And those are the ones that are kind of juicy. There's a lot of water in them, good to eat. You know, we also talked about dry fruits, which tend to be dry when the seeds are ready to be dispersed, right? We also did a comparison between apples and oranges. Remember that? We did that, and we found out that such a thing can be done, right? And we also went into tropical and subtropical fruits, okay? So today we're sort of going to continue on this track, but we're going to go into a little bit different topic, even though it is, you know, related for sure. We're going to get into seeds, okay, as well as some other things, all right? So first of all, okay, our objectives for today. We're going to first look at another subject of interest, the oldest germinated seed. It's amazing, you know, this is going to be amazing. Okay, and this is documented. Next, we're going to go a little bit into the structure of seeds and the components of a seed and into germination. Okay, what is germination and what happens during germination, okay, of the seed. Then we're going to do a little reprise on dicots and monocots. If you recall, you have certain kind of plant structures that are characteristic of dicots, and we also have certain kinds of plant structures that are characteristic of monocots. Okay, the thing is that it's not true in all cases, okay, just like in any other science, whether it's chemistry or physics or others, what you want to do is you want to memorize the generalities and then you try to memorize the exceptions, okay, because there are always exceptions to the rule, okay. Lastly, we're going to talk about plant hormones because, of course, plants are living things and we animals, we have hormones too, right, like epinephrine, you know, all these other kind of hormones. The one I'm most familiar with is epinephrine, right? Anyway, so that's a stress hormone. Anyway, but we're going to talk about plant hormones too today, okay? So in terms of epinephrine, a lot of you students must be used to a lot of stress. This is sort of like par for the course for students, you know? And here at Leeward Community College, there's even a workshop, I think a connection workshop that they call Stress for Success. So if you can, that might be a good idea to go to some of these, you know, workshops, and because it can give you really a lot of good benefit for the time that you invest in it. Just like anything else, school, just like work, just like your home life, the more time and effort you invest into it, the more you'll get out of it. Yes? Anyway, so plant hormones as well. Alrighty, so we're going to look at the oldest germinated seed, and this is documented. This is scientifically documented, okay? And this is a plant called the sacred lotus, also known as Nelumbo nucifera. And here's a beautiful picture of a beautiful flower, okay? And this sacred lotus, the oldest germinated seed, was found several years ago, quite a few years ago, and they could date it because what they did is that they'd done archaeological excavations of this lake bed in China. And what they did, you know, when you do archaeological excavations, you scrape, you know, one layer and another layer. Well, in one particular layer, which was like over 1,000 years old, you know, what they did is they found the seed, and they actually had a horticulturist or an agriculturist, somebody who knows how to take care of plants, to germinate it. It was amazing, okay? So they actually had a plant that came from this germinated seed. So we might think, you know, seeds, you don't sometimes think of it as being alive, but they really are alive. Yeah, you have a little embryo inside of the seed like we saw last time in that peanut that we looked at, okay? So you might think of a seed as an inert, passive, you know, not even living thing, but it is living. It's actually going through the process of cellular respiration at a very, very slow rate. And in the case of this sacred lotus, okay, it was actually doing this respiration for over 1,000 years, okay? So if we can kind of go back to the here, so here, you know, when we were looking at the flower, okay, this part here, which is a fruit, of course, develops from the ovary, because you remember a fruit is a ripened ovary. And what is the ovary? It's one of the structures or part of the pistil, and the pistil is the feminine or female part of the flower. So here we have a fruit of the lotus, and if somebody is taking it apart, here's a seed. Okay, so it is actually a plant, a flowering plant. You saw the flower earlier that develops into a fruit, and then a result of that is a seed. All right, so they did find a seed of the sacred lotus. Not just that, is I think people know also that you can actually eat lotus, yeah? I think people do this in Asian cuisines. They call it lotus root, and they call it hasu, and then they use them in all kinds of different kinds of recipes, such as nishime and other kinds of Japanese, as well as Chinese, and a lot of different Far Eastern cuisines use lotus root. And another thing about the lotus too, you can see here, okay, the lotus is actually a plant that is very symbolic. Okay, so the seed itself was more than 1,200 years old. Amazing. So that was how they dated that particular strata of the lake bed that they excavated in. Okay, so they were able to compare, you know, the seed where it was with when it was washed into the lake. Because when you have these sediments washing into these bodies of water, you'll have a good chronology. You have an idea what is earlier deposit and what is later deposit. The more toward the top of the sediment something is, the younger it is, or that, you know, it was deposited less than the ones under that, the layers under that. Okay, so this sacred lotus seed was from a deposit from more than 1,200 years old. And another thing about the lotus, you see that it's called the sacred lotus. It is symbolic to Buddhists. Okay, so what happens is that if you actually have a lotus flower, the thing about it is that the lotus, of course, is an aquatic plant. Its roots are in the mud. A lot of times the roots are actually growing in the mud, but it has that beautiful flower, you know, that we saw before. And so this is sort of something that Buddhists kind of thought of, you know, in terms of somebody being able to express their best nature, is that your feet are on earth or your feet are in the mud. It's dirty, you know, the world sort of pollutes you, but at the same time you yourself have the potential to blossom into the most beautiful flower that's possible to you. Okay, and usually when you see a statue of Buddha, yeah, when you see a statue or a scroll paintwork of Buddha, even at the Honolulu Academy of Arts, they have this great collection of Buddhist art. And whenever they show a Jizo statue or some of the other Japanese Dainichi Nyorai and like that, what you'll actually have is you'll have the image standing on or sitting on a stylized lotus, because they figured that the Buddha, which is supposed to be a really evolved being, you know, much more than, you know, what we call a person or a human being or a man, but generally, you know, they think that the person, it really is elevated to such a height they can stand on or sit on only something as pure and as beautiful that comes from the mud as a lotus blossom. Isn't that interesting? Okay. All right, now the seed itself. Okay. The seed is composed of three parts. We have, first of all, the seed coat. That is the outermost part of the seed. Sometimes it's hard. Okay, sometimes it's almost impermeable to water. That means water can't actually diffuse or osmose, okay, into the seed so easily. Okay, this is why sometimes when you have seeds, like from cold weather areas or from other places, sometimes you need to do something called scarification. Okay, so scarification, what does that mean? That means you get a little razor, you have your seed, you nick it with a razor perhaps. A lot of native Hawaiian plants need to be pre-treated in this way. Or you could do something like have the seeds in a coffee mug, boil some water, pour the water over that, let it sit, and then what that'll do, that'll actually disrupt the seed coat a little bit. Another thing that people have done, I've done it myself when I worked over at the Nitrogen Fixing Tree Association, is what we did is we actually had the seeds of Lucina leucocephala, which is the koa haole, you know, which people think is a rubbish plant. We actually harvested the seeds, and what I had to do when I had a whole bunch of them is to pour pure sulfuric acid on it. Can you believe it? Let it sit for like about 5 minutes or something, pour it off, and then that actually softened the seed, you know, so you could actually, you know, have germination happen. So seeds, the seed coat is a wonderful protectant of the seed. It allows a little bit of gas exchange, allows a little bit of oxygen to come in, carbon dioxide to go out, because you remember those are the gases that are involved in cellular respiration. Okay, so oxygen is used and helps to break down a larger compound, and then what you'll get is ATP. So respiration happens in these seeds at a very, very slow rate. Witness, this lotus seed that somehow was kept alive, in all this mud, entombed in mud for over 1,000 years. Okay, so the seed coat, a very important component of the seed. It helps to protect the embryo that's inside, as well as it allows the interaction, the interface between the outside environment and the environment inside of the seed. Yes? Now another component of the seed is known as the embryo. Okay, the embryo itself is a little tiny baby plant, and it's actually a baby plant that's inside. Okay, so the three components that make up every seed, just about every seed, is a seed coat, the embryo, and then you have the endosperm. Recall that the embryo is the individual organism that develops from sexual reproduction, right? You have the egg and the sperm, right, coming together in the process known as fertilization. And then after that happens, you have a diploid or 2N, or 2 sets of chromosomes in a zygote. When the zygote undergoes mitosis and develops into the embryo, okay, that is actually what happens. You have a sperm and an egg resulting in the development of the embryo. Then you'll generally have the sperm and the polar nuclei. I know your reading talks about that. The endosperm could be triploid. It could be, have 12 sets of chromosomes, maybe in some cases, but generally this endosperm is very, very nutritious. Okay, so the endosperm, the job of the endosperm is to serve to give nutrients, okay, to the embryo, to keep the embryo alive, however long that the seed is, sort of like in a dormant state. Okay, that's when the seed is just lying there, when it doesn't necessarily look like it's really alive or anything, but seeds are very much alive generally. Okay, so the seed coat, the embryo, and the endosperm. These are the parts of the seed, generally. There are very few exceptions, such as orchid seeds have very little or even no endosperm. This is why orchid plants will actually, make a fruit, and they'll have up to 1 million seeds in a fruit. And part of the reason for that is that they don't have endosperm, so you can crowd 1 million seeds into that. And orchids have that kind of life cycle. They actually give a lot of offspring, and very, very few actually germinate and actually grow into adults that can produce more things. So this is like one way a life cycle of a plant can go. Can you actually produce lots of young, lots of them die, and you only have a few to grow to maturity. Okay, so that is also exemplified in biology in terms of animals. Zoology, such as the elephant, has very few, and even us, when we compare ourselves to some insects, they'll lay hundreds of eggs sometimes, Anyway, so we're not as prolific, but we tend to take care of our young for a longer time. So we have a different kind of life history than other kind of animals that produce lots and lots and lots of eggs or lots of young, and then just leave them to fend for themselves, high infant mortality, and etc. Okay. All right, so here's a nice diagram of the seed. So this one here is maybe about 1 millimeter. Remember, a millimeter is 1,000th of a meter. So a centimeter is about less than ½ an inch, right? Something like that, a little less than ½ an inch. And if you actually have the seed, this is about, let's see, ½ an inch. And then what you'll have is 1/10 of that. Okay, so this is what is being depicted here. And you can see several structures. First of all, you have the outermost part of the seed that we talked about a little bit. And this is called the seed coat. Right, the seed coat, and its job is to serve as an interface between the embryo and the rest of the seed inside and the outside environment, as well as it serves a protective function. Like if an animal has eaten a seed, such as a bird, what will happen is that it'll go through its gravel, right? The bird has a crop that allows it to break down some material. So there are actually some seeds that need to go through an animal's digestive system and be either treated with the acid in their stomach or being, mechanically abraded by the crop in a bird in order to germinate after the animal has excreted it outside of its body with a little bit of fertilizer, right? So we have the seed coat. We also have, in some cases, you might call it the endosperm, but in this case, the endosperm has gone to form, it has given the energy to form the cotyledon, okay, in this case. Remember what a cotyledon is? A cotyledon is the seed leaf. Okay, seed leaf or 2 seed leaves of a particular plant, of a particular flowering plant. The cotyledon will sustain the embryo, after the seed germinates, and we'll get into that in a little bit, but the seed coat will actually split open, and then we'll have a bunch of miracles happen, okay? Now this here is what they call the hypocotyl. This is the area below the cotyledon, and this is actually going to develop into the radicle. What is a radicle? The radicle, and here it is spelled correctly. This is how you spell it. Okay, it doesn't end with C-A-L, but it's a little bit different spelling. But this radicle is like the embryonic root of the plant. So this is actually going to, when the seed coat splits, what's going to happen? Okay, we're going to talk about germination in a second. Is that the seed coat will actually swell up, imbibe or take up water, and the seed coat will swell, and then finally it'll split. And what will happen is this little radicle will leave this environs here and grow, and then as we're going to see in a little bit later, we're going to see a cotyledon. So we'll kind of take a look at that next. But first of all, let's do a little bit about dicots and monocots. This is going to be like a little bit of a reprise, okay, sort of like a repeating what we had before, okay, for one thing. Okay, so remember that dicots, they have two cotyledons, and a good example of a dicot is a bean. Okay, do you recall what kind of root system dicots have? Okay, just think about it for a minute. Okay, so here we have a dicot. You can see has two pairs of leaves. These leaves here, these are the cotyledons. This seedling is probably only maybe an inch tall, if that. Okay, so a couple of centimeters tall. So here, okay, so here's a cotyledon, here's another cotyledon. So these are called the cotyledons or the seed leaves. And that is what lends its name to dicot. Remember, DI stands for two, di is two, and cot refers to cotyledon. So this one here has two cotyledons. And now when you look at this here, these are the true leaves. Generally, in a lot of plants, the true leaves look different, okay, from the seed leaves, all right? So in this case, this one has two of the seed leaves, and it has two true leaves, what they call true leaves in it. And these leaves would be more characteristic of the plant as it matures. So do you recall what kind of roots, okay, the dicot has? Okay, remember we have tap roots in dicot. So here, in this nice photograph, we have a main root, okay, coming off the stem. So generally in a dicot that germinates from a seed, you'll have a tap root. Okay, and here you can see the smaller side roots coming off of it. And if you were to look at a cross section of a typical dicot root, this area, excuse me, this area around this area here, these would be parenchyma cells. Okay, so this would kind of make up the cortex of the root, parenchyma cells, and interior to that you have the vascular cylinder with the endodermis and the pericycle. Remember that? We also here have this little cross shape, sort of cross shape area, and this, of course, do you guys remember what this is? Yeah? This is xylem vessel cells, xylem vessel and also tracheate cells, okay, in the root. That generally in a dicot root, you'll have this sort of configuration if you do a root cross section. And then this area here, the areas between the wings or the outposts of the xylem are where the phloem is. If you look closely at it, you'll see larger cells with smaller cells next to it. Remember, you have the sieve tube members and the companion cells, right? The sieve tube member being bigger and being actually the one that will load the sugar or actually be transporting the sugar, and the companion cells actually helping and being a companion to the sieve tube member and loading the sieve tube member with sugar juice, okay? All right, now another thing about dicots, okay? Remember what kind of vascular bundle arrangement that you find in the stems of herbaceous dicots, or dicots that are not woody, or dicots that are woody but in their first year of growth? Well, you have this kind of vascular bundle arrangement where you have the vascular bundles all arranged in a ring formation, okay, on the cross section of a stem. This part is called the pith, more large parenchyma cells here, okay? And if you look closely, you can actually see these cells here. If you look closely at them, these are collenchyma cells, which help to function in support of the stem. So remember collenchyma and sclerenchyma and parenchyma, three different kinds of cells in ground tissue, right? So we have parenchyma, fairly large cells, a lot of it taken up by vacuole. Okay, tend to have only primary cell walls, and they're kind of like a general cell of the plant. Collenchyma, okay, with unevenly thickened cell walls, they also tend to be only the result of primary growth, and they sort of have a cobblestone effect. And then in some cases, such as some xylem cells, you'll actually have sclerenchyma. okay, where you have fiber cells. Remember that fiber cells are part of the xylem, part of the phloem in the secondary growth. Well, then you have secondary growth in those cells, right? And so you'll have these fiber cells, okay? Now, do you recall what kind of venation in leaves, okay, that they had? So here, a really nice leaf, and if you look closely, you can see that the main vein, the midrib here, you have the secondary veins going off of it. Do you remember what kind of vein venation this is? Yeah? Remember that this kind of venation is called pinnate venation, where the secondary veins come off the primary vein like the pinnae, or like a feather, yeah, the parts of a feather. Yeah, so we have netted venation because this one does have pinnate venation. We also have palmate venation, but generally in here you also have the secondary veins, you have tertiary veins, quaternary veins, and it's all forming a bunch of networks, okay, inside of the leaf. Okay, so dicots tend to have netted venation, and the netted venation could be either pinnate venation or palmate venation. Do you recall from what we saw what kind of flower parts and in what numbers? Yeah? Okay, just kind of think about that. I won't give you the answer to that. You have to think about it, okay? You can be sure that this is going to show up on an exam now, okay? All right, now we also have monocots. So dicots, dicotyledons, monocots have a single cotyledon. Okay, a good example of a monocot is corn, all right? Remember too that corn is like wheat or like rice, it's a grain. Remember that grains, you actually have the embryo and the seed itself, you know, sticking to or actually being connected to the fruit. So a corn kernel is not a seed, it's actually a fruit called a grain, where you cannot dissect out, you know, the seed apart from the fruit, all right? So remember monocots, and in the case of this one, we have corn, okay, being a good example of a monocot. Right? Do you remember what kind of roots? Okay, so here, this is a picture of somebody growing, you can see their hand here, they're growing something hydroponically. What does that mean? It means they're actually growing a plant in a liquid medium. They're not actually planting it in the ground, but they put it in a nutritive medium that has, you know, nitrogen, phosphorus, potassium, and other macronutrients as well as micronutrients that the plant needs in a small amount. So do you know that there are people here on Oahu who are growing vegetables hydroponically? Not just that, is that they're actually growing crops, tomatoes and such, using hydroponics, and they can actually keep their crops kind of disease-free and such. And did you know that there's also a hydroponic store out in the Kahala area, across from Kahala Mall? Okay, I had a student who actually works there, and so that was good. You know, so hydroponics, it can be a very good way to grow plants. So anyway, so getting back to the question, you have these fibrous roots where the roots are all about the same size. So they form like a bunch of fibers, yeah? So fibrous roots in monocots. Recall too that if you look at the cross-section of a monocot root, here you have the xylem. So you can see that it looks really different from the dicot, yeah? So here we have the monocot xylem, and in this area between them, this part here that is colored sort of like an aqua color, it happens because, not because the cell walls are staining this, are actually this color, but because the person who prepared the slide actually had to stain it. They actually had to probably slice it up in a cryotome and then stain it with, you know, 2 or 3 different stains. And so that's why you can see things so well when you look at a prepared slide, because a lot of the work of making the slide has been done for you. If any of you go on into botany, okay, there's a class called Plant Anatomy at UH. It's really a wonderful class. The teacher will actually teach you how to make slides, you know, how to stain them, and it's really, really interesting. I think when I took it, the Plant Anatomy, we had the use of something called a cryotome. What is a cryotome? Have you ever heard of cryogenics? Remember what cryogenics is? It's when somebody has passed away, you know, like that kind of hippie guy, what was his name? That he died, and he had them cut his head off after he died, and it's in storage. Can you believe that? That's kind of wild, huh? Anyway, so cryogenics means that you freeze a person or part of a person in the hopes that in someday, you know, far off into the future, they can be actually resurrected, which I don't know, it sounds kind of iffy to me, but still. What is his name? Timothy Leary. That was the guy, yes. Anyway, so cryotome, it's like a big machine. We can actually put a piece of plant in there, put it in there, and what it does, it freezes it instantly. It freezes it because what you have is a super sharp razor that will come and slice it really, really thin. Because remember that with a compound light microscope, the light has to be able to show through the specimen. So in this way, using a tool like a cryotome, you can actually make slices thin enough so that you can look through them, or the light can move right through them. It's really, really fun. So if anybody is interested in cells or in tissues, take plant anatomy or even animal anatomy. Very interesting, Okay, now here we have another cross-section, or a section of a cross-section of this looks like a monocot. Remember that monocots have scattered vascular bundles, so in the whole area, okay, of the cross-section, you'll actually have vascular bundles all scattered along the whole areas of the section. Okay, so monocots have scattered vascular bundle arrangements, okay, in their stems. Then lastly, well, not lastly, penultimately, okay, almost lastly, we have these leaves. So this clearly is a monocot. You can count the flower parts, right? Count the flower parts. And generally here, we also see that the leaves have parallel venation in this case. So in this case, we have veins, secondary veins and tertiary veins probably, that run parallel to the midvein of these leaves. Another good quality of monocot leaves that you can look for is that oftentimes monocot leaves tend to be a lot longer than they are wide, right? Not in all cases, but in some cases. Okay, any kind of clue that you can get, to use is a good thing. Okay, now generally this lily here, 1, 2, 3, 1, 2, 3. I can't really tell. This one, I think, and this one are the sepals. Okay, you can see that the sepals in this case are colored the same color as the petals. 1, 2, 3 petals. So this, of course, is a characteristic of a flower of the monocots. They have flower parts in threes, okay, or multiples of threes. Okay, so this one happens to have 3 sepals, 3 petals, probably 10 stamens or 6 stamens, I think maybe 6, yeah, in lilies. Okay, anyway, so you can kind of tell. So one of the things that you will probably be called upon to do in lab is to be able to identify whether a flower or any part of the plant, okay, is from a dicot or from a monocot. Okay, that's very important because this is a good way that you can make qualitative differences, between a dicot and a monocot. This is the great divide, okay, in the angiosperm group, in the angiosperm phylum, or what we used to call division. Okay. Okay, now getting into seed germination and development. We talked a little bit about scarification. Well, we do also know that seeds or plants, okay, oftentimes, okay, they'll oftentimes be in cold areas where it would not be very conducive for the seed to germinate under, adverse conditions, yes? So the seed coat actually will protect what's inside in order to allow the seed to survive this kind of adverse conditions, whether it's fire, whether it's frost, okay, whether it's some kind of other upheaval, all right? So generally what the seed needs, it needs to have enough moisture in the atmosphere that's in the environment that's available to it. It also has to have an availability of oxygen gas. Remember, oxygen gas is needed for the seed to undergo cellular respiration or aerobic cellular respiration, and you also have to have a certain kind of temperature. Okay, so generally in plants that germinate and live in temperate areas where you have a really distinct winter and summer and fall and spring, okay, generally after the seed has overwintered, maybe frozen in the ground, and then what will happen then is as it warms up and you start to get spring, you'll have new things germinating. So what is germination? Germination is merely the sprouting of the seed. It's when the seed coat split and the radicle starts to emerge. Okay, that's germination, and once that happens, the plant is then known as a seedling, S-E-E-D-L-I-N-G, okay? Okay, so the radicle emerges, and that becomes the root of the plant, okay? Another thing, too, is that once a radicle leaves the seed, it can start absorbing water, okay, more, because, of course, the seed will first start imbibing or absorbing water in the seed coat, which will allow the seed coat to split and the radicle to come out of the seed coat, okay, the radicle germinating, and then what will happen, the radicle will absorb water, right? Another thing that happens is that the shoot starts to emerge. What is the shoot? Remember, the shoot is the stem and the leaves of the plant. Okay, so the shoot emerges and then it'll green up. So here we have a whole bunch of beans, talking about being full of beans, you know. So here you have we have lentils and you have all different kind of beans. And here you see that the root or the radicle has emerged, and some of them even have a little bit of the shoot. Here you see the cotyledon, okay, greening up in some cases. So generally, when the cotyledons first come out, you know, when the seed germinates and the root generally doesn't have any pigments in it, so it's not green, right? So what'll happen is that the shoot will emerge, you know, it'll be sort of like this, with maybe this being the root, and it'll do this sort of thing, and then it'll do that. And then what happens is that when the shoot first emerges, it too is like a creamy whitish color, no pigments present. But as it's exposed to light, what will happen is that the chlorophyll will actually manifest itself. The cells will actually make organelles called chloroplasts. You remember chloroplasts from a long time ago? Right? Those are actually the organelles of photosynthesis. So what happens is that in the cotyledons or in the seed leaves, it'll green up. Okay, it'll actually manifest itself. It'll actually make chlorophyll. Okay, in the chloroplast. Remember, chloroplasts are plastids. The generalized plastid type, you know, a protoplast or something like that. I'm just kidding. Well, what will happen is that it'll actually become a chloroplast, and you'll have the chlorophyll developing, and then you'll have it greening up. Okay, and then when that happens, then the cotyledons are starting to photosynthesize, and then the plant can now grow even faster. Okay, because then the plant doesn't have to rely only on the stored reserves in its seed leaves, because that's the function of the seed leaves or the cotyledons, or in the endosperm. Okay, it can then make its own food now, because what they have is a food reserve that was provided them by the double fertilization, right, by the endosperm. Okay, now what we're going to talk about next, okay, is about chemical messengers, right? These chemical messengers are often known as plant hormones. What do? Okay, for one thing, a characteristic of a hormone, just like in our own bodies, okay, generally hormones are effective at very, very, very low concentrations, such as parts per 1,000, parts per million, usually parts per million or parts per billion. So even if the plant or the animal is only making low concentrations of these hormones, as long as the body is balanced, as long as everything's going fine, pretty much the body of the plant or the animal can carry on with what it needs to do in order to survive. Okay, just like in our bodies. Okay, U.S. students, I think I talked about this a little bit before. U.S. students, you know, generally are stressed out a lot. Okay, this is like part of a course of being a student. Everybody experiences that when they're a student, because generally one of the things you're pressed for is time. A lot of people, they have families and they have their job, plus they have school. So that's like, you know, sort of like a triumvirate of things that rule over you, you know. So the thing too is that what you want to do is you want to be able to do everything, but of course it's very difficult because we only have finite time, and there are consequences to be had if you consistently sleep 3 hours a night for 5 years. You know, that's going to take a toll on your body, you know. Anyway, so what happens is that when we're under stress, when we humans are under stress, we actually secrete more of what we call adrenaline, also known as epinephrine, from the adrenal glands that are located sort of on top of your kidneys, you know. And it actually provokes in people a whole bunch of reactions, and some people can distill it to fight or flight. Things like you're holding your breath, you know, and that kind of stuff. So even if your adrenal glands are only putting out a little more epinephrine than usual, it actually will cause a little bit of an imbalance, and then so you have a lot of stress syndromes, you know, because your body is saying you're in danger, you know, you're stressed out, you know, you're actually in danger, you could actually, you know, be killed or eaten, you know, because this is our bodies, you know, our bodies, we're animals. Anyway, so generally, even if you have a little bit more of hormones, that will have a big effect on your body, you know, including things like exam time, you're feeling really hungry all the time. It's because you're stressed out. Your brain, your neurons are firing much more than you know, would happen is if you're at rest or not stressed out, you know. So so this will have actually an effect on your body, as well as a drop of epinephrine that happens, you know, after the stress is gone. After finals are over, you know, I used to go to the university, you know, student, they have a health clinic on the campus of the University of Hawaii. Okay, and very nice doctor that I know there, she said that 2 weeks after finals, there's a whole bunch of students and faculty that come in 2 weeks after finals, is because the stress hormones in your body, which are at a kind of high level, will actually keep you going through finals week. Just like when you go on a trip, if you go on an exciting trip and you're having a lot of fun, stay up half the night and all this kind of stuff, going to coffee shops and doing too much reading and just gallivanting around, what happens is that your stress hormones are up, so you're up. You can actually be in Las Vegas and be gambling all night, all this kind of stuff. But what happens is that about the week after or 2 weeks after you come back, you crash, right? So this is the same physical phenomenon that this doctor had seen, medical doctor has seen at the clinic. 2 weeks after finals, there's a whole bunch of students in there. It's because they're catching and actually manifesting in their body all the germs that were around them all the time. What usually happens is that microbes, bacteria, fungi, viruses, they're around us all the time. They're in the air, they're on every single surface that we touch, they're on our bodies, they're in our bodies, but what happens is that they're opportunistic. It's when your resistance is down, which happens when you're as an aftermath of stress, then the germs that are there can actually then invade and cause symptoms in your body. And the symptoms that you usually get are usually because your body is fighting them off. Uncomfortable symptoms such as mucus, such as coughing, such as inflammation. These are your body's responses to being invaded, excuse me, by these different microbes, etc. Okay? Anyway, so getting back to plant hormones, okay? They're chemical messengers, and they tend to be effective at very, very low concentrations, even in plants, okay? Usually counted in parts per million or parts per billion even, right? Another thing too, another thing about these chemical messengers is that generally, like the epinephrine in our body, being produced by our adrenal glands and then moved to various parts of the body, they're usually made in one place and then they're translocated to someplace else where they have their effect, okay? That's another sort of a description of a hormone. Another thing about hormones, as we know, is that you have growth and development. Like usually, when you say your children, or maybe even you, when you were, just entering into puberty, one of the things that they'll say is that somebody's starting to get hormonal, because what happens is that when you enter into puberty, you have the production of a whole bunch of hormones, male hormones and or female hormones, both actually, because both men and women have a little bit of the other sex's, primary hormone, right? Anyway, so hormones function in growth, okay, and also development into adult stages, as in, in animals. Okay? So one thing we're going to talk a little bit about, okay, is the first hormone we're going to talk about. I think that we have 5 that we're going to talk about. And the first one in terms of plants is something called auxins. Okay? Auxin is actually a kind of plant hormone. It's like a classification of plant hormones, but there are a whole bunch of different kinds of auxins. And this is actually a diagram of indolacetic acid, also known as IAA. Okay, it's a kind of auxin, right? It is among the more common of the auxins, okay? Now, these auxins, okay, are kind of famous because amazingly, Darwin had to do with the development and actually the manifestation of this, okay? So generally, Darwin, okay, and his son, we're going to see a picture of them in a second, okay? Let's see. All right. So Darwin and son, which you're going to see in a minute, okay, actually were among the first workers, okay, that first worked with these auxins. And this auxin is usually made in growing parts. What does that mean? At the tip of the stem, at the tip of the root, okay? Auxins also have a great role to play in something called cell elongation, or the maturation of tissues, okay, from an immature small state into a larger state and more mature, okay? Just like we saw in the root, where you have the area of cell division and you have the area of elongation, right? Another thing auxins can do, they can actually inhibit the development of buds, and they also have a role to play in the growth of what we call adventitious roots. Do you remember what adventitious roots are, right? So adventitious roots are, of course, they're roots that, remember, they actually develop from places in the plant that you don't expect the roots to develop from, such as on the stem, okay, or even from a leaf. Okay, so that can happen, Now another thing that plant hormones or this auxin has to do, and this is a very interesting thing, they have a role to play in something called parthenocarpy. Did we talk about that before, parthenocarpy? Okay, think about the word. What do you think it means? First of all, you see that carpy, that refers to carpel. Remember what a carpel is? A carpel is a folded over leaf, because remember with a flower, all is blatt, right? It's all leaf. So it's actually a folded over leaf that has seeds inside. Okay, that's what a carpel is. And you can have one or more carpels making up a pistil. Okay, partheno, what does that refer to? Does that remind you of any word that you've seen? Have you ever heard of the Parthenon? The Parthenon in Athens? It's like a beautiful ancient Greek building. It's called the Parthenon, right? So it is actually a place that is in Athens. And Athens in Greece is the capital of Greece, and it's actually a city that their Greek goddess that was, you know, the ruler or the overseer of Athens was Athena. Athena is the Greek goddess of wisdom, okay? And she was also a virgin goddess, okay? She was a virgin goddess, and she was actually sort of conceived interestingly in that her mother was Zeus. He was one of her girlfriends. She was one of his girlfriends, and he actually found out that any male child that she would bear him would be greater than he was. So he didn't want that to happen. So when she was pregnant, he swallowed her. Okay, and then what happened then is that the baby developed, and then he complained of a terrible headache, and then one of the other gods, Vulcan, I think it was, cracked open his head with an axe, and Athena sprang forward from his head, full grown, you know, uttering a battle cry or something like that, because she's not only the goddess of wisdom, but she's also the goddess of war. Okay. Anyway, so parthenocarpy means that you actually have a flower or you know, a sexual structure on a plant, but you don't have fertilization happening. What you have happening is that you have a fruit developing, sometimes without seeds, okay, but without benefit of fertilization. Just like a banana, you know, like you have a banana. Almost everybody's in Hawaii has eaten bananas, right? If you peel it, you bite into it, those little black things in it, those are ovules. Those are unfertilized ovules. So banana fruits actually develop, okay, from unfertilized flowers. Isn't that interesting, right? Also things like, you know, what kind of fruit? I think they call them persimmons, which Japanese people like, you know. They're very good tasting actually, very high sugar content, but they too, if you buy a persimmon like Christmas time, you know, the ones maybe that aren't too expensive, you know, to make a fruit basket or something, if you bite into them when they're ripe, lots of sugar, no seeds at all. So auxins can be used for initiating flowering as well as initiating fruiting without the benefit of fertilization. Isn't that amazing? So you have fruits, you know, with no seeds, right? Anyway, so here, this is the picture I was looking for, okay? So you might think one is Darwin and this one is Charles Darwin. Okay, you might think that this is his son. That's not true. Of course, this is a portrait painted of Charles Darwin. Look at him. Doesn't he look like a very intelligent fellow? Oh, he has that nice high forehead and everything. Anyway, so this is Charles Darwin, and we're going to find out more about him when we talk about evolution, okay? He was named as one of the most important people of the last 1,000 years, the last millennium, one of the most important, influential people in the last millennium. Okay, so this is Charles Darwin, and as a young man, and this is his son, Robert Darwin. Okay, no, Francis. I'm sorry. His name is Francis Darwin. So anyway, when Francis Darwin was a young child or a young man, and Charles Darwin, you know, he was already a settled married man. And then what they had done is they'd done these experiments. Okay, so they were actually the people who first did work that showed that there were auxins that were present. Okay, so what we're going to do is we're going to talk about that a little bit. So what they figured, you know, Charles Darwin, he was fascinated with all kinds of things having to do with plants, with animals, with the natural world, about, you know, natural history. He just loved it. You know, ever since he was a little kid, he was out collecting shells, collecting fossils, collecting plants, shooting birds and collecting them, you know. So one of the things that they did is that Charles Darwin wondered what happens, okay? What happens when you have a plant that has light coming only from one direction? Right? The plant actually bends over this way and starts growing in the direction of the light. So Charles Darwin and his son Francis were really, really intrigued by this. They were thinking, what happens with this? So what they did is they actually did little experiments, you know, they had this little shoot tip, like a little shoot tip like this, and they covered it with a little hood. Okay, and then when they did that, the plant did not bend toward the light. So Charles Darwin and his son Francis Darwin came to the conclusion that, or maybe there's some sort of substance that is produced by the tip of the stem that, you know, makes the plant bend toward the light. Remember we mentioned that before too. It's called phototropism. Okay. Now later workers did further work on this. Okay, so if we can kind of go to the elmo here, okay, and then what we'll see is that if we have a little stem, they call it coleoptiles, right? Say this is a stem, and then what happened is that another worker actually cut off the tip of the stem and then tried to grow the plant this way. And of course, if the light was coming from this direction, the sunlight, whatever light's coming from this direction, okay, like this, okay, so it didn't bend with the tip of the coleoptile gone. Another thing they did is they actually took the little stem tip here, like this, put it on a block of gelatin that had been infused with chopped up pieces of a stem tip. You know, what they did is they actually allowed this tip to diffuse its whatever substance it had into the gelatin. Then they put a little bit of this gelatin or this gel up on top of that, not the coleoptile tip, but a piece of the gelatin. And then what will happen? It actually started bending toward the light. Okay, so later workers feel that with the coleoptile, I know these are very primitive drawings, but anyway, as long as they get the message to you. So what happens is that, okay, so you have the fact that there's some substance that this coleoptile tip made or gave off that caused this stem to bend toward the light, whereas one that didn't have any of whatever substances was did not bend toward the light. Nowadays, people think that generally, if you have a stem bending toward the light, generally you have the coleoptile, right, the tip of the stem, and then what happens is that they think that here at the tip, you have some kind of a substance being produced, okay, that will actually move to the opposite side. So if the sun's shining this way and then the plant starts bending this way, it's because whatever substance is being secreted here is actually moved from this tip to this side of the stem, makes this side of the stem grow longer. So then what happens in comparison, this side of the stem is shorter. So what happens is the tip bends toward the light. So in terms of phototropism, or when the plant is actually growing to the light, okay, they believed, and they actually found these experiments that probably it was true. There was some substance that was produced at the very tip of the stem that caused more growth on one side. Okay, so Darwin and son, you know, Charles Darwin and his son Francis were the ones who first actually looked into that. Interesting, huh? Now, generally, auxins are made in actively growing parts of the plant, whether it's the tip of the shoot or the tip of the root. So here, I wanted to show this as an example of made in actively growing parts. So something like a big pine or a gymnosperm, it's very strongly have what they call apical dominance. That means generally the tree will just keep on growing taller and taller. That means the meristem, remember meristems? Right, this is the place of actively dividing cells, okay? So the meristem tip, somewhere here at the top of the tree, apical dominance means at the apical meristem, it will tend to just keep growing taller and taller and taller. Okay, what happens if you actually cut off, you know, just like these guys cut off the tip of the coleoptile? What if you cut off the stem tip? What will happen? Well, the hormone that was made, the auxins that were made at the tip of the stems, aren't there anymore. So what happens is that another effect of auxins is that, okay, it tends to inhibit the growth of lateral shoots or lateral growth. Remember that when you have a stem, you have places where leaves were on the stem when it was younger, and these are places where you also have buds, right? You have the lateral buds or axillary buds. When you cut off the tip, the actively growing part, that will actually allow for side branching to happen. Just like in certain plants, like hedges, you know, like mock orange. What happens if you have a mock orange hedge in your front yard, you know, to keep any of passersby from looking into your home? What happens? Well, if you're the person taking care of the yard, whether you're the son or the daughter or the mother or the father, what happens is that when you hedge it, you use your hedge trimmers or you use your pruners, you cut it back, that actually makes the plant grow more bushier. Okay, so that what you're doing is you're actually putting the fact that these auxins aren't being produced to actually allow for more side growth. So you get more bushiness, you get actually more side shoots coming off. So people who trim their hedge over and over and over, they'll have a nice thick hedge, right? Doesn't that make sense? So auxins usually made at the tips of roots, tips of shoots in actively growing parts. And here we have another example of phototropism. What's happening? You have light coming from this direction, you have this plant growing toward the light. So these are three different roles. We have growth in a lengthwise direction. You have the removal of apical dominance in this case, so when auxins are not being produced, you actually have these guys coming out to the side buds. And of course, these tips then are going to produce more auxins. Okay, so with a hedge, you have to keep trimming them back all the time in order to make sure that it keeps on coming out bushy, all right? And it also has a role in phototropism, okay, a growth toward the light. Another function of auxins is that it does function in cell elongation, just like in the example I just showed you with the coleoptile tips. Okay, so when auxin is made at the tip of the coleoptile and is translocated to the other side of the stem away from the light, where the cells on that side, on the dark side of the plant, will actually grow longer. Okay, as well as like that. So it has a function in cell elongation as well as in cell division. All right. Another thing about auxins is that generally auxins will tend to inhibit the formation of buds or inhibit the growth of buds as well, such as you see here. Here we have a couple of flowers. They look sort of like roses, or are they peonies, something like that. So some kind of a temperate area flower, maybe a camellia, not sure. Anyway, so I'm not really up on the temperate flowers very well. But here you can see that the auxins will kind of inhibit the development of buds, okay, whether it's vegetative buds or floral buds. Okay, they have a tendency to do that. Another thing that auxins can do is they can provide for the growth of adventitious roots. So if, say, if you're working at a nursery or if you're at home, you're a plant person, you like to actually make more plants from one plant, okay? Even if you have a hibiscus hedge, you can actually make cuttings, you know, off healthy parts of the hibiscus and dip the tips, you know, in something called a rooting hormone, which is composed of partly of auxins. Okay, that will encourage roots to come out of the stem. Okay, so those of course are adventitious roots. So you can oftentimes have really good propagation results. A lot of native Hawaiian plants, generally, if you can get about 50% of the cuttings to root, that's a very, very good percentage, okay? And usually that is helped along in commercial nurseries as well as in nurseries that serve the public in terms of making new plants. So you can actually stimulate the growth of these adventitious roots if you have a certain proportion of auxins, you know, around the root, the place where you want the roots to grow from, such as a stem. Okay? And auxins also have a role to play in parthenocarpy. Not just that, is auxins can also induce flowering. Okay? One of the things that people do in pineapple fields, right? Other places, apple orchards, places like that, they'll usually have auxins or something like that, and they'll give the plants a whiff of this auxins. Okay? And so they'll spray them with auxins, and what that can do, that can actually allow for the formation of fruits, okay, from these flowers. They will allow them to actually have a whole bunch of fruits fruiting at the same time, because one of the biggest expense, you know, in a botanical garden or in an orchard, any place where you're growing plants, one of the biggest expense, just like every place else, is your personnel, you know. So what's going to happen if you have a whole orchard of plants? What you want to do is you want them all to flower and fruit at the same time. So if you're going to hire people to come in and pick your fruit, they can actually come there all at one time within 1 or 2 weeks and pick all the fruits that are there. You don't want them coming back, you know, every week for months at a time, because so this is one of the ways that we humans have utilized plant hormones. We actually make synthetic ones, and we'll actually spray them on plants, okay, to allow for parthenocarpy. Or parthenocarpy is like when you have the development of fruit without the effect of fertilization, okay? And then here's another example too. Auxins have also been used as herbicides. What is an herbicide? Herb means plant, and side, just like homicide, means that you've killed a person, which is very bad, of course. If you have herbicide, you actually kill plants, okay? So this is actually a crop dusting plane that's flying over a jungle area. Okay? Auxins include things like some of the compounds that were used in what we call Agent Orange, okay, which is a defoliant. Okay, so Agent Orange has actually been used to kill other plants, as well as there have been allegations too that a lot of people who were in the war in Vietnam, that they were exposed to this Agent Orange. Okay, so it's dangerous to humans as well as plants. That's my conclusion anyway. So auxins can also be used as an herbicide. See, the thing about these plant hormones is that they're a little bit difficult, okay, because some of them You know, we're just talking about auxins right now, but there are going to be 4 others that we're going to talk about, okay? So generally, these different plant hormones will have different effects depending on the concentration that you actually put of this hormone. You can put it at low concentration, it'll have an effect of inducing flowering. You put it at a different concentration, it'll actually have an effect of allowing leaves to drop. So some of these different hormones actually have effects. So some of them will actually work counter to another, and some of them will actually enhance the effect of another hormone as well. So hormones are not an easy thing to pin down, you know, as some of us might think. Now, another kind of hormone that is very important in the development of plants and etc. are called gibberellins. Okay, it was first discovered, okay, by here we have the nice structure of it too. Okay, beautiful animation here. So I have gibberellic acid, Ga. Okay. And so these gibberellins, okay, are responsible for something called foolish seedling. It was discovered by a man named Yabuta. Okay, Yabuta, he was a Japanese scientist, and what it was, he first observed it in these rice seedlings that were infected with a fungus. So isn't this gorgeous image? This is actually, I think, an aerial photograph of rice paddies, terraced rice paddies in China. Okay, so he observed these in these dwarf rice plants. So the plants actually grew really tall and skinny. They shot up and they grew up so tall that they would fall over and die. Okay, because they'd fall over, injure themselves, and then die. So the guy called it bakanai. Baka means fool, right? So that meant foolish seedling. So he said that they had foolish seedling disease. Okay, so then the plants grew very tall. In fact, that there's an image which I haven't been able to get a hold of, but there's an image of a man standing on a ladder holding onto this really tall rice plant, maybe 12 feet high. Okay, so this hormone will actually have an effect in bolting, in elongating the stem to actually make the stem really tall. Okay, all right, so these gibberellins also. Okay, so what we have is the stem elongation of plants that are usually dwarfs. Okay, so if you have a plant that comes out really small genetically, just because that is a habit of it, that's how it's supposed to come out. If you actually apply gibberellic acid to it, it will cause this bolting or sudden elongation of the stem. It can take a few weeks, but you can have the elongation of the internode. Remember what the internode is? The area between the nodes of a stem. So you have stem elongation of dwarf plants. So this is actually a picture of very tall conifer trees. Yeah, isn't that awesome? Another too is that gibberellins can also promote the germination of seeds. So here we have individual fruits of sunflower. This is actually a fruit, and the seed, of course, inside. Let's see. Let me think about that. Yes, these are actually fruits called achenes. Okay, the seed itself would have been the part of the sunflower that you would eat. Okay, that is actually the seed. Yeah, and of course, most of what you're eating when you're eating a sunflower seed is that you're actually eating a lot of endosperm, right? And also partly the cotyledons as well. Okay, because remember, endosperm is a really, really important part of our diets, you know. Okay, another thing is that these gibberellins will also cause a bolting of what they call biennial. So this, of course, is broccoli, right? Did you know that broccoli and cabbage and let's see, cauliflower and kale and Brussels sprouts and other more exotic things such as kohlrabi, these are all the same species. They are all Brassica oleracea. Okay. Anyway, so these are also biennial plants, you know, just like cabbages. Okay, etc. So what happens is that in these biennials, okay, in these biennial plants, you'll actually have, okay, the plant, you know, putting up. You remember when we talked about the phloem, we talked about biennials. We talked about in the first season, you have the germination, the growth of the plant, putting out the leaves, photosynthesizing, making a big taproot, right? And then when the winter comes, the top part of the plant dies, the root stays in the ground, which is why, you know, places in the Far East, daikon and other root crops, carrots and such, keep well over the winter time because there's a lot of food reserves in there. Well, what happens in the second year of growth, the food from the root will go up into the stem, making more leaves and making a big flower. So biennials actually have a cycle of producing their flowers and fruits after 2 years, biennial. Okay, 2 years, over 2 years of growth. So, so what can happen is that if you have a plant of some kind, a biennial, that's actually putting out, you know, its leaves in the first year, I think you can actually cause a bolting and flowering of it if you apply gibberellic acid. Okay. Okay, now another plant hormone, okay, is called the cytokinins. Okay, cytokinins, of course. Okay, here a nice structure of a cytokinin of some kind. Okay, cyto, of course, means cell. And what cytokinins can do is it can stimulate cell division. Okay, so this is just a reminder, you know, of different kinds of cell division that we talked about a little bit about binary fission, yeah, about what bacteria go through, and mitosis and meiosis, which plants and fungi and animals and protists go through too. Okay, because we are of course made of eukaryotic cells. Okay, so cytokinins can stimulate cell division. So cytokinins and all these other plant hormones, including auxins, are very, very important in terms of tissue culture. Yeah, what is tissue culture? Is when you actually take a little part of a plant, or animal even nowadays, you actually can take a little bit of part of a plant, whether it's from the leaf, whether it's a root, whether it's from the stem, you can actually put it in a little test tube plants, and actually allow it to proliferate. So if you add a little bit more of cytokinins, you can allow the undifferentiated cells to keep proliferating, making more and more and more cells. And then what happens is that in a growth chamber, you know, a sterile environment, as sterile as you can make it, the worker will actually get this callus. Remember the callus that we saw on those adventitious growth roots that we showed you, the adventitious growths? Okay. What happens is that this callus, or undifferentiated tissue, you can actually take it, cut it apart, plate it out on either a petri dish or in a flask on a good nutrient media that has cytokinins in it. And what it'll do, it'll actually produce more and more cells. It'll actually continue to divide. Okay, and then what can happen is that if you then transfer it into another flask with auxins and other some such, okay, you can actually allow for the growth of roots in the plant. So you can actually program these cells to actually grow or not grow, depending on the kind of hormones that you apply to it and whatever other growth factors are in the media. Another role that cytokinins play is in differentiation, okay, in terms of the particular general cell type, like parenchyma cell, making up, you know, the different organs of the plants or the different tissues of the plants. So cells will actually differentiate, okay, from a generalized cell type to a more differentiated cell type and specialize for certain functions, okay? And here, differentiation. We're showing you differentiation here in animals, okay? Even though we're a botany class and we are talking about plants, but still, this is a good example showing about differentiation in a particular kind of animal. So some of you may know that as, you know, the zygote, the single-celled result of the fertilization event, sperm and egg coming together in an animal, generally what will happen, you'll have many events of mitosis, forming something called a blastula, okay? A blastula, which is like a hollow ball of cells. Well, then that will then eventually develop into something called a gastrula, okay, a gastrula stage. So generally, almost just about all animals go through these stages as it's developing, okay? So here you start off with a zygote, okay, which has two sets of chromosomes, a single cell, and then what happens is that it develops into a blastula and a blastocyst. Okay, then you have the development of a gastrula. You see the word gastrula, gastro. What does that make you think of? Gastronomy, okay, gastric ulcers. Oh my gosh. Anyway, so this has to do with your digestive system. Okay, so animals generally, we have a digestive system because we need to eat our food, right? Okay, so what happens then is that we have the development of this. You have an external layer. Okay, and then you'll have different kinds of cells developing from that. So here we see that this generalized cell type, okay, from the ectoderm is actually going to develop into different types of cells, whether it's a neuron, okay, whether it's epithelial cells, okay, or other cells like that, right? Then we also have in our bodies too, or in what will develop into what we call germ cells that will eventually either give rise to egg or give rise to sperms. Okay. Then we also have a middle layer, okay, which also differentiates into other kind of tissues. Okay, I know animal cells or animals are very much more complicated than plants, but this is a good example of differentiation where you have generalized cell types differentiating into specialized types. So we have muscle cells, whether skeletal muscle or cardiac muscle or smooth muscle. We have red blood cells. We'll have white blood cells of various types. We'll have platelets. We have different kinds of other kind of epithelial cells. Okay. Now, so we'll kind of go through these. So the internal layer too, developing in different kinds of cells as well. Okay. Anyway, another effect of cytokinins is that you see these leaves are beginning to senesce. What does senesce means? You think of the word senescence, you think of the word senile, the senile dementia. We have all these cheerful things that we're talking about today. Anyway, so senescence is like the aging and the eventual death of the cells, okay, or of the organ. So here, this plant's chlorophyll is actually, breaking down and leaving it. And this is a leaves that are actually turning red because of the fact that you have the carotenoid pigments that are there all the time, but generally since the chlorophyll pigments aren't there, not so much on these leaves here, only on this area here, then you can see the underlying carotenoids, okay, pigmentation. Now another hormone, okay, is called abscisic acid. Okay, and here we have a beautiful structure here. Chemistry is a wonderful subject, I think. Okay, so what does abscisic acid? They called it abscisic acid because they believed that it had to do with abscission, or when leaves are ready to fall off the plant, had to do with senescence and abscission. But what they found out is that it doesn't really have that much of a role to play in abscission. Okay, so abscisic acids actually have to do with dormancy in seed, allow seeds to actually, and buds to remain dormant until the weather is good enough and conditions are good enough for them to develop and grow. Another role that abscisic acid has in terms of closure of stomata, this is sort of theoretical, but people think that abscisic acid does have a role to play in the closure of stomates. Okay, so here's some nice pictures of stomates. Okay. Now the last hormone that we're going to talk about. So before they thought that abscisic acid had to do with abscission, but they found out that is not the case. They found out, in fact, that it is actually ethylene that has more to do with abscission and senescence. Okay, so here we have here on the screen here we have a nice depiction of the chemical structure of ethylene. Very simple molecule. It just has 2 carbons and 4 hydrogens. The 2 carbons double bonded to each other. Because remember, carbon likes to make 4 bonds, right? So each of the carbons have 2 hydrogens bonded to it plus a double bond to another carbon. Okay, and we have a space-filling molecule here too. Okay, on the slides. Okay, right here. Yeah, so here you see the 2 carbons and the 4 hydrogens. Okay, so this is ethylene, okay, also known as ethene. So ethylene gas. Do you know that when you're kind of walking down the hall, every time you brush past a plant, every time you touch it, it actually has wound tissue to it. And generally, if you wound a plant, even inadvertently, or sometimes, you ever have a plant on your desk and somebody comes and starts playing with a plant on your desk, picking off the leaves and, pulling it apart, things like that. Tell them, leave your plant alone, because what's happening is that the plant is actually making ethylene gas as a result of being wounded. Okay, because if you're actually tearing a plant apart, that's wounding it. And here on the slides, we can see a beautiful bunch of ginkgo trees. Okay, so these here are actually senescing. The leaves are actually, chlorophyll breaking down. And did you know too that an abundant or too much of auxins can actually promote the development of ethylene too. A high amount of auxins present in a plant and being given off by a plant will actually cause ethylene to be produced as well. And here we have beautiful, fall foliage, So hard to really believe it sometimes, but generally, even the end of a season can be a beautiful thing as well. Okay, ethylene gas can also cause fruits to ripen. Anybody ever told you, hey, if you have some mangoes and they're not quite ripe yet, put them all in a paper bag, and then what they'll do is they'll ripen, because what happens is that ethylene gas causes fruits to ripen. So if you have like a bunch of green bananas, like a hand of bananas, maybe 6 or 7 bananas, because of course a banana bunch really is big, So anyway, so if you have bananas that are still green, they say, oh, put it in a plastic bag, tie a rubber band around the mouth of the bag. What will happen is that the bananas will actually ripen faster because you have ethylene gas all contained in there, okay? So if you want to get your pineapples to do that too, just like what I was talking about with auxins, they'll actually give them a whiff of auxins. High auxin will actually promote ethylene production. If you actually give a whole bunch of pineapples a whole bunch of ethylene gas, they'll all ripen at the same time, which is what they want in commercial fruit production, you know? Another things they'll do is that they also convert starches in the fruits to sugars. So you ever eaten a green fruit, either sour or they're really starchy? You know, like, of course, lemons, like these green lemons, they'll be really, really sour. And I know lemons, like regular ripe lemons, so these green lemons will actually color up, and they'll actually do that. They'll actually apply, ethylene to fruits to make them color up, because we humans, we like things that look nice. Even if it's the best kind of lemons you can ever get, if they're all warty looking and if they're all bumpy and they have all these marks on them, you usually don't want to buy them, yeah? Even if they give you the biggest amount of juice, okay? Now, apples and oranges are also given whiffs of this ethylene gas, especially oranges, to make them have that nice orange color that helps to sell them in the market. Okay, look at that. Isn't that nice? Doesn't it make you think of summertime? Can you imagine a picture of some kind of citrus fruit juice with all these slices floating around in that? Isn't that nice? Yeah? Okay, and then now another thing too, we're going to kind of think of the world's largest fruit. Okay, so generally they hold it to either be pumpkins or squashes. And then I kind of saw this debate, you know, on the internet, and then they said pretty much it's either pumpkins or squashes, just like comparing apples and oranges as well. Okay? Okay, anyway, that's what we have time for today, and we're going to summarize a little bit, okay, about what we want to talk about. We talked about the oldest germinated seed, and we also talked about seed structure and germination as well, very important parts in the life of a flowering plant. We did a little reprise on dicots and monocots, and we also covered some plant hormones. Anyway, so that's all we have for you today, okay? Hoping that everything's going well with you, and let me know, okay, what's happening. We thank you, and bye-bye.